Hello future doctors. So in today's class, let's find what's right or wrong and let's bring the statue to listen heart's song. When you see the shining moon, the Luna, you listen dub. When you close the doors of your heart, you fall in love. So with this prop, I'm gonna start today's brand new class, that is body fluids and circulation. Yes, my dear future doctors, I know exams are coming near day by day and you people are getting restless, heart is beating quite faster. Now, I'm here to give a boost towards your preparation, that is the journey, the aim, all about NEET 2026. And myself Biswajit, your zoology mentor, and I'm assuring you, after completion of this particular lecture, you will feel confident enough to solve the questions of the chapter. If we talk about the topic ECG, after completion, you are gonna say it's very easy, G. If we talk about the topic cardiac cycle with proper explanation diagrams, I have broken that whole topic from NCERT into step-wise manner, right? So, without delaying, start the chapter, and we don't have to be worried about the curated questions. Proper sets of questions, TPPs, and the handwritten notes. You just have to click the link given in the description box, and that will lead to a new world, that is PW app. The access of batches you will be getting there only, and I'm there with a full series of NEET Zoology where I'll be covering each and every chapter of class 11th and class 12th, right? So let's start. So my lovely Zuzu gangs, for this chapter we'll be covering all the topics from NCERT, starting from the introduction and the body fluids, that is blood, and the assistance of the blood, that is filtered blood, lymph or tissue fluid. We'll be studying about circulatory pathways, double circulation, we'll be talking about systemic circulation, pulmonary circulation, and how cardiac activities are regulated. We know that there are two types of hearts are present in animals. One is myogenic, one is neurogenic. We'll be differentiating among them, and we'll be concluding the chapter with few disorders related to cardiac system or circulatory system. You know that most of the times, most of the students, they are confused that, sir, what is the difference between heart attack and heart failure? Everything will be cleared after watching this video. Okay, so let's start with the introduction. Now, before starting that, just try to imagine the transportation system of a particular city. So it has three important components: transportation vehicle, roadways, and petrol pump to fill up that transporting vehicle, right? Similarly, our circulatory system, inside our body, we have three kind of similar kind of systems, right? One, the transporting vehicle, they are blood and lymph, right? They are acting as the mean or the media to transport the gases, transport the nutrients, transport the excreta. And for the flow of the blood, we have blood vessels, and you need to generate force for that particular flow. So for that, Almighty has given us a biological machine that is called blood pumping station of our body, heart. So if you are asked that what is the importance or need of transportation or circulation, then you have to answer that there are two important necessity. First necessity, the transportation of the circulatory system of our body, they act as delivery guy. That delivers nutrients and oxygen to each and every cell. And secondly, they help in removal of unwanted material like carbon dioxide, which is the end product of the cellular respiration, and obviously the excreta, which we call the metabolic waste. So basically, to give backup to the metabolic activities, we need to supply some good materials to the cells, and we need to eliminate out some bad material from the cells. Who will be performing this responsibility? Our respiratory system or transportation system. Now, if you observe animal kingdom, there are different types of animals are present. Few are ancient one, undeveloped or quite simple body designed one, and few are advanced one. They have complex body design. So the animals which have quite simple body design, they don't have specialized circulatory system, so they are performing water-driven circulation. Like you can observe among poriferans like sponges and coelenterators like hydra and jellyfish. That's because their body design is quite simple, right? But what about more complex organisms like us? Our body is made up of billions, trillions number of cells. So we have specialized body fluids. One, the primary one, that is blood, and there is a helping hand or the middleman of our body, that is lymph. So blood and lymph, they are acting as body fluids for complex organisms, right? So here the mechanism of circulation is shown through this diagram of a sponge. Now if you observe this, you can observe the sponge is intaking water with the help of numerous tiny pores which we call ostia, and eliminating out that same water with the help of a large pore, that is osculum, and continuously it's creating a water current inside and outside. And while this water is flowing through inside and outside the body, there is exchange between oxygen and carbon dioxide during the time of the circulation of water through the canal system inside the body of sponges, as well as they are eliminating out the excreta. They are eliminating out the excreta into that water. So the purpose of transportation is solved. Okay, and eventually when the water is carrying food particles with it, there are specialized cell, they will be capturing or trapping the food particles, right? Now let's talk about our cardiovascular system. That means we have a heart, we have blood vessels, and inside those vessels blood is flowing through. So first we are going to talk about our primary body fluid, that is blood. Okay, now by origin blood is mesodermal in origin. So blood is mesodermal in origin, just like other connective tissue. Blood is red colored body fluid. So it's written here, blood is a special connective tissue. Now why we are considering blood as special connective tissue? What is making it difference from other connective tissue? See, there are three important components you need to make connective tissue. Some cells, and those cells will be producing fibers and ground substance or matrix. Now for special connective tissue, the ground substance will not be produced by the cells of the own tissue, and in case of blood the fibers are also absent. So that's why you can consider blood as a fibrous tissue. So that's why we call that blood is a special connective tissue consisting of fluid matrix. Now what do you mean by this fluid matrix? In blood, fluid matrix will be produced by liver and spleen, we call it plasma, and blood has its own cells or fragment of cells, we call them formed elements, right? So you can see here the major two components of blood, plasma and formed elements. Blood is mesodermally originated, like in our embryonic developmental stages. If you observe our embryo, we had three layers: outer one ectoderm, inner one endoderm, and the middle one mesoderm, right? Now why we call blood as a false connective tissue? We call blood as false connective tissue as because the ground substance or the matrix or the fluid matrix of the blood isn't produced by the blood cells, it's produced by some other organs like liver and spleen. That's why you are calling blood as false connective tissue. The mechanism of blood production, that is called hemopoiesis or hematopoiesis, the process of formation of blood. Okay, the organs which are responsible for formation of blood, we call them hematopoietic organs. And the primary organ for hematopoiesis in our body, there is our bones. There are certain hematopoietic bones, and inside the bone you will be getting yellow bone marrow, you will be getting red bone marrow. Now, specifically this red bone marrow, that is considered as hematopoietic organ. So what are the organs where hematopoiesis takes place? Red bone marrow, of bones, also liver helps in this process, as well as spleen. Remember the organ spleen, that is we call it graveyard of RBC and the blood bank of our body. Spleen is also called as blood bank of our body because RBC production also will be taking place inside the spleen. Okay. Now for the process of hematopoiesis, we need some special hormones. We are gonna like elaborate in detail in some future slide. So these are the hematopoietic organs. Also hematopoiesis is called hemopoiesis, right? Hem means blood in Latin language, and inside an adult human being, you will be getting near about 5 to 5 liter of blood is thriving inside our blood vessels, right? Now, if you take out 100 ml of blood from a person, and if you want to observe what are the components present inside the blood, so you are drawing that 100 ml of blood inside a test tube, and you need to keep that blood like for 15 to 20 minutes undisturbed condition. You cannot disturb this test tube. Now after that, what you are going to observe? You will be observing that 55 ml of the blood is floating upwards and that become colorless. We call that plasma. And 45% or 45 ml of the blood, the solid components sedimented downwards. We call them formed elements along with RBC, WBC and platelets. So basically 55% of blood, the volume of blood is contributed by plasma, and 45% of blood volume is contributed by the formed elements or by the blood cells and platelets, right? Okay. Now in detail with the table, we are going to learn different components of blood. So just now we were discussing that blood has 55% plasma and 45% formed elements. Now what about plasma, the ground substance or the liquid matrix of blood, which is produced by liver and spleen. Plasma is produced by liver and spleen, right? And the color is straw colored and viscous fluid. Where plasma is produced inside liver and spleen and then comes into the blood flow, right? Now what are the components of plasma? You will be finding that the maximum volume of plasma is actually water, 90 to 92%, and remaining 8 to 10% are different solid components. They are primarily plasma proteins, some minerals, nutrients and hormones. Okay. Now 6 to 8% you will be getting presence of plasma proteins. There are 3 important plasma proteins are present into blood: fibrinogen, albumin and globulin, right? Okay. Now if we talk about fibrinogen, these are the largest plasma protein, help in blood clotting, acting as one of the clotting factor. Blood has or the plasma has 13 clotting factor. So this fibrinogen is one of the clotting factor. Now let's talk about the plasma protein albumin, the most abundant one. Highly rich, plasma is highly rich with albumin, and that helps us to maintain the osmotic pressure or osmotic balance of our body. In detail you are going to learn in the chapter of excretory products and their elimination, when we will be detailing about kidneys. Then really we are going to learn about how albumin is helping us to maintain our osmotic pressure inside our body, right? Remember, they are the most abundant protein. Largest fibrinogen, most abundant albumin. And what about globulin? There are 3 types of globulins present inside the blood. alpha globulin, beta globulin and gamma globulin. Now specifically this gamma globulin, they are very much important for us. They help in our defense mechanism. We call these gamma globulins as antibodies. And there are different types of antibodies. We are naming them as, remember the word damage. So IgD, IgA, IgM, IgG, IgE. So these are the gamma globulins present into our plasma. And if you ask, sir, what is the function of alpha and beta globulins? They act as carrier proteins of iron and copper. Okay. Now, what about the minerals present into the plasma? Sodium, chloride, magnesium, bicarbonate, calcium and very little amount of potassium. Okay. In plasma, more amount of sodium is present, right? As well as chloride ions, magnesium ions, bicarbonate ions, HCO3 minus, calcium ions and very little amount of potassium ion. Okay. What about the nutrients present? Glucose, amino acids and lipids are also present in transit form. Means blood is acting as a delivery guy, right? So whenever blood is taking those nutrients, dissolving into the plasma, and then after some time they're gonna deliver it to some particular tissue or particular organ, right? That's why they're present in transit form. Their level is always changing into the plasma. And also plasma includes some hormones, right? Plasma includes some hormones as well. Now a question is, very important question is asked, what is serum? Now serum is plasma minus clotting factors. Plasma minus clotting factor equals to serum, the colorless fluid. After we are getting any wound and after the blood is coagulated near to that wound, still after a few minutes, a few seconds, we can observe a colorless fluid is oozing out through that. So that colorless fluid, which doesn't have the components of clotting factors, we call that serum. Plasma minus clotting factor equals to serum. And what about the formed elements of blood? And why they are named as formed elements? As because they are formed by bone marrow and then thrown out inside the bloodstream. So that's why we are calling them formed elements. They are formed inside the bone marrow, right? There are three types of formed elements are there: RBC and WBC. We call them corpuscles. We call them corpuscles. They are intact cells. And platelets, they are broken piece of cell. They are broken piece of cell. So platelets, the meaning of platelets is broken part of cells. Okay. Another name of RBC, erythrocytes, because they are red in color. Another name of WBC, leukocytes, colorless or whitish in color. It is Latin name. Another name of platelets, thrombocytes. They help in the process of thrombosis or blood clotting. That's why another name of platelets given thrombocytes. Right. Let's move to the next part of our video. We're gonna talk about formed elements. Formed elements, they are formed inside bone marrow, rather red bone marrow. Okay, so different cells are given here. So this is RBC, acidophil, basophil, neutrophils. There's certain types of WBC, macrophage, monocyte, lymphocytes. There are also certain type of WBC, and the broken piece of the megakaryocyte cell, that is platelets. So one by one, we're gonna study in detail about them. So first, let's talk about the RBC. What is the other name of RBC? Erythrocyte. Okay. Now the process of formation of erythrocytes is erythropoiesis. And for erythropoiesis, you need some special hormones like erythropoietin. You need cortisol. You need thyroid hormones, as well as you need testosterone hormone. So these are the hormones which are responsible for hematopoiesis, or more precisely for erythropoiesis. Where this erythropoiesis takes place? Erythropoiesis takes place inside the red bone marrow, RBM. Erythropoiesis also takes place into the liver, spleen, and in your life cycle, the first erythropoiesis is observed during the time of embryogenesis, and it was by the yolk sac. Yolk sac is one of the extra embryonic membrane or sac present outwards the embryo during the time of our embryogenesis. The first erythropoiesis is observed, or first RBC was produced inside our body by the yolk sac. Then the RBC will be produced by liver and spleen, and right now in our body primarily RBC is produced inside red bone marrow. And the hormones which are responsible for this process: erythropoietin, a hormone produced by kidney, particularly juxtaglomerular cells of kidney and the mesangial cells of kidney. Another hormone is cortisol, help in the process of erythropoiesis. Another hormone, the hormone from thyroid gland, thyroid hormones. And also the prime male sex hormone or the androgen called testosterone. This hormone also helps us in erythropoiesis. That's why we'll observe that the more the number of RBC, the more the volume of blood. So if you compare the blood volume in male and female, okay, a male or males have the higher volume of blood in compared to female. In a female, you will find that 4.5 to 5 liter of blood, and in a male you will find 5 to 6 liter of blood. Okay, now why there is extra volume of blood present in male is because, see, erythropoietin, cortisol and thyroid hormones present in both the sexes, but as because males we produce more amount of testosterone, that helps us to boost up the production of RBC as well as blood. Okay, what is the number of RBC? 50 lakhs near about, that is 5 to 5.5 million per millimeter cube blood or per deciliter blood, right? This is the number of RBC into our body. So 5 million means 50 lakh per deciliter blood or per cubic millimeter blood. Okay. Now what is the average lifespan of RBC? Average lifespan of RBC, we know everyone knows that this is 120 days. Now why 120 days? We know that there is a process we call the maturation of RBC. RBCs are considered as enucleated, matured RBC. So when the process of maturation of RBC with the help of the hormone erythropoietin is happening inside the red bone marrow, the erythroblast, the precursor cells of RBC, we call them erythroblast, will be eliminating out nucleus, eliminating out mitochondria, will be eliminating out the other important cell organelles like endoplasmic reticulum, even Golgi complex. And after that, it will become enucleated, and the one and only motto of that RBC is to accommodate more and more amount of hemoglobin molecules. We'll find that near about 35% of the volume of RBC is occupied by hemoglobin molecules, and they help in the process of transportation of oxygen and carbon dioxide. That's why RBCs are red in color, and if we talk about the shape of RBC, they are biconcave or dumbbell shaped, right? Especially we are talking about the mammalian RBC, right? Now, I was telling that hemoglobin, the main motto of mature RBC is to accommodate hemoglobin. So 100 ml of blood contains 12 to 16 grams of hemoglobin. You need to memorize this information. 100 ml of blood contains 12 to 16 grams of hemoglobin. Basically what happens, 1 gram of hemoglobin, 100 ml, and that 1 gram of hemoglobin can carry 1.34 ml of oxygen, and ultimately that hemoglobins, they helps us to form oxyhemoglobin. Basically they are the carrier molecules of oxygen, right? And spleen is known as graveyard of RBC. Why spleen is known as graveyard of RBC? Graveyard means upon the expiry, when the RBCs are getting expired, their size will become larger, right? And the special kind of reticulate or reticular tissue present inside the spleen will trap them while the RBCs are passing through. Basically, every time the spleen, they are doing screening of RBC, and when they are expired, they will trap the abnormal RBC or the expired RBC, and they will call macrophages, and macrophages will break them down, right? And after that, we'll see there is formation of bilirubin, biliverdin, and the iron is released. So that's why the destruction of abnormal and the expired RBC after 120 days happening inside the spleen. So that's why it got the name the graveyard. As well as remember that spleen is also called the blood bank of our body. Not only destroys the RBC, it can produce the RBC as well. Okay. Now we'll be talking about WBC. Now before talking about WBC, I just want to tell that there are certain abnormalities with respect to RBC. So if there is less number of RBC, what do you call that condition? If there is more number of RBC, what do you call that condition? Okay. If there is less number of RBC, we call that condition erythrocytopenia, or anemia. And if there is more number of RBC present into our blood circulation, that is called polycythemia. Okay. So you need to remember this particular terminologies. What? Erythrocytopenia or anemia. If there is less number of RBC. If there is more number of RBC, there is polycythemia. Now let's talk about WBC. We call them leukocytes. Now the process of formation of WBC. The process of formation of WBC, we call it leukopoiesis. We call it leukopoiesis. That also takes place inside the red bone marrow with the help of some particular factors and hormones. You may call them the leukopoietins. Okay. The number of WBC into our bloodstream, there is nearby 6,000 to 8,000 per deciliter or per cubic millimeter blood, or per deciliter blood. There is a number of WBC. Average lifespan of WBC varies from 10 hours to 10 days. 10 hours to 10 days, right? Okay. Now there is some abnormal situation of WBC that also can be observed. One situation that is called leukemia. Another situation that is called leukocytosis. Another situation that is called leukopenia. Now, which situation is called leukemia? Leukemia means abnormal rise of WBC in blood. That is called also can be called as blood cancer. Leukemia, leukocytosis, more WBC in compared to the normal one, that is 6,000 to 8,000 per deciliter blood. If there is more WBC in your blood circulation, then that condition is called leukocytosis. Generally, that is observed after any kind of infection in our body. Okay. Now, what is leukopenia? In case of certain diseases, the pathogenic organisms, they will kill our WBC, and then there will be decrease in the number of WBC count. So that condition is called leukopenia. Decreased number of WBC. Leukopenia. Okay. Now types of WBC. If you observe the WBC, there are few WBC. Inside them you can observe enzymatic granules. So we call them granulocytes. In some other WBCs where you cannot find that these WBCs are with some enzymatic granules. So if you are observing a particular WBC where enzymatic granules are present, we call them granulocyte. If you are observing a particular WBC where the cytoplasmic portion is very much clean, there is no granules present, we are calling that a granulocyte, right? Okay. Now there are three types of granulocyte. presents basophils, eosinophil and neutrophil. There are two types of agranulocytes present, monocytes and lymphocytes, right? Okay. Now, the very important questions are asked regarding their population in our blood. Okay, if you are asked that which is the most abundant WBC of our body, then you have to answer neutrophil. Their population in our blood is 60 to 65%. Okay, which is the rarest WBC in our blood? You need to answer basophil, 0.5 to 1% population only. Okay, second most abundant WBC in our body, second most abundant WBC, that is they are lymphocytes. Third most abundant monocytes, 6 to 8%, and fourth abundant 2 to 3%, that is the population of eosinophil or acidophil. Okay, now these three WBC, basophil, eosinophil and neutrophils, why they are named so? Basically they are colorless, and if you want to observe under the microscope, you need to stain them, you need to color them. Okay, now when you are giving color to this particular WBCs, these basophils, they will absorb only basic stain. Eosinophil, they will absorb only acidic stain, that is eosin, and neutrophils, they can absorb both the acidic and basic stains. So depending upon their absorption capacity of stain, WBCs are named so. Okay, now what is the function of basophils? See, basophils, they release three important chemicals inside our bloodstream. They are heparin, responsible for they act as the anticoagulant, responsible for keeping the blood fluidity inside our body. Okay, histamine, act as vasodilator and bronchoconstrictor agent. They can decrease the diameter, increase the diameter of a blood vessel and decrease the diameter of our trachea, responsible for allergic reactions. And serotonin, they act as vasoconstrictor. So these three important chemicals produced by basophils, heparin, histamine and serotonin. Again I am repeating, function of heparin is acting as anticoagulant agent inside our blood. So that's why inside our body blood is not getting clotted or coagulated. Histamine, it has two important functions, acting as vasodilator and another is acting as a bronchoconstrictor. Dilation means increasing the diameter of a pipe, constriction means decreasing the diameter of a pipe. So histamine, vasodilator, serotonin, vasoconstrictor. Okay, and this histamine and serotonin, they are responsible for producing inflammatory reaction just after any kind of infection or allergic reaction. Okay, eosinophils, they resist inflammation. That's why they are also responsible for, I'm talking about eosinophils or acidophils, they are also called anti-allergic WBC. They will be producing some enzymes that will break down the histamine, which is the prime allergen. So antihistamine enzyme, okay, will be produced by eosinophil. Also sometimes eosinophils can produce allergic reactions. So the prime WBC, which is responsible or associated with allergic or anti-allergic reaction, you need to answer they are eosinophils. They can be both anti-allergic and allergic. Okay, and also another most important function of eosinophil, they attack the multicellular pathogen. When suppose ascaris, you are suffering from ascariasis, you are suffering from teniasis, when the multicellular pathogens are attacking you, especially the nematodes or platyhelminths are invading into your body, then the soldiers which will be getting activated, they are eosinophils or acidophils. So they also act against multicellular pathogen. Okay, what about neutrophils? Neutrophils, they will be attacking unicellular pathogen. They have the phagocytic activity like amoeba. They can engulf the pathogen by the process of phagocytosis, the cytoplasmic streaming. I was talking about the neutrophil. They will attack the unicellular pathogens, right? And they are phagocytic in nature. Okay, so they act like amoeba. Now these basophils, eosinophils and neutrophils, if you observe their nucleus, what you can see, that their nucleus is just irregular in nature, not like the conventional shape of nucleus they are having, right? So like in basophil, you can observe they have a headphone shaped nucleus. In neutrophil, their nucleus is of 3 to 4 lobes. So multilobular nucleus you will be finding inside basophils, eosinophils and neutrophils, right? So that's why we call them polymorphonuclear leukocyte. All these three, basophils, eosinophils and neutrophils, they're also, especially these granulocytes, you can call them polymorphonuclear WBC. Okay, again I'm repeating, for basophils you can find trilobed. For basophils, trilobed, 3 lobes of nucleus can be observed. It can be of S shaped. For eosinophils, is the nucleus is bilobed or headphone shaped, and for neutrophil the nucleus is having 3 to 4 lobes. So that's why multilobular nucleus. But for a granulocyte, monocyte and lymphocytes, they have normal nucleus. Monocytes have nucleus like kidney shape. The nucleus is just like kidney bean. And for lymphocytes, the nucleus is quite large, and sometimes you can observe cartwheel shaped nucleus, right? Now let's talk about monocytes. Monocytes, they are very large cell. They are the largest WBC. Monocytes, they are the largest of WBC. Well, they are circulating into the blood, we call them monocytes. But when they will be entering into the tissue, they will change their shape with larger pseudopodia, then we call them macrophages. So macrophages and monocytes, basically they are same cells. In bloodstream we call them monocyte. In tissue, they are responsible for attacking again the unicellular pathogens along with the neutrophils. By the process of phagocytosis, we call them macrophage. So that's why if someone asks you, name the phagocytic WBC. So there are 2 phagocytic WBC, monocytes and neutrophils, right? Monocytes are also called macrophages while they will be invading into the tissue. Now let's come to the lymphocytes. There are primarily 2 types of lymphocytes: B lymphocytes or B cell, T lymphocytes or T cells, right? Now they are responsible for forming our adaptive immunity. Basically, B lymphocytes, why it's called B lymphocytes? The first time the discovery of B lymphocyte was inside birds, and in birds there is a lymphoid organ called bursa fabricius. That's why from bursa fabricius it's getting the name B lymphocytes, the letter B. Now the T lymphocyte, from where we are getting the letter T? T for thymus. The training school of T lymphocytes, the site of maturation of T lymphocytes, is a primary lymphoid organ inside our body, that is thymus. So both the B cells and T cells, they will be produced into bone marrow and they will be matured into the thymus. From thymus, T lymphocytes got the letter T. B lymphocytes, they are responsible for producing antibodies. That's why that immunity is called humoral immunity, because the antibodies are present produced by B cell, but they are released into the humor or plasma of blood. So that's why immunity by B lymphocytes, we call it humoral immunity. And the immunity given by T cells, they are just like commandos of our body. They will be directly interacting with the pathogen, and either they will be directly killing them or they will be helping other cell to kill the pathogens. So the immunity we will be getting from T cells, we call that cell mediated immunity. Okay, now under a granulocyte there is a third type of cell. NCERT is saying about that into a different chapter. The name of that cell is NK cell, natural killer cell. Natural killer cell. That is also special type of a granulocyte, right? Okay. Now let's talk about platelets, the thrombocytes. The process is called thrombopoiesis, and for that you need the hormone called thrombopoietin. The number of platelets into our body, that is 1.5 to 3 lakhs per deciliter or per cubic millimeter blood, right? There is the number of platelets, and average lifespan of platelets varies from 7 to 10 days. Now, what is the meaning of platelets? You know that there is the broken parts or broken pieces of cell. So basically, during the time of this process of thrombopoiesis, you will observe there is a large cell with a large nucleus that is called megakaryocyte cell. And upon maturation, the megakaryocyte cell will get degenerated and the broken pieces will be forming platelets. That's why we consider these are platelets are not true cells. Platelets are not true cells. So if someone asks you, name a cell which is not a true cell into our blood, then you need to answer it is the thrombocytes or platelets, because during the time of formation of platelets, we got a cell with large nucleus that is called megakaryocyte, and upon the action of the hormone thrombopoietin, that will be forming some fragments, and those fragments or broken pieces of megakaryocytes, we call them platelets, right? So platelets, they can release a variety of substances, most of which are involved in the coagulation or clotting of blood. So platelets will be releasing some clotting factors, right? Without platelets, there will be no coagulation taking place. A reduction in their number, if there is less number of platelets into your body, can lead to clotting disorder, can lead to blood clotting disorders, which will lead to excessive loss of blood from the body. Specifically, you will be learning that platelet will be forming platelet factors or the endothromboplastins, which are initiator molecules for the process of blood clotting. So if there is less platelet, you will be suffering from clotting disorder. And what do you mean by clotting disorder? If there is blood is reluctant to clot, produce clot, excessive loss of blood from your body will be there. Okay. Now, let's start a new topic, that is blood grouping. Now, this is gonna be a very conceptual topic. See, there was a scientist called Carl Landsteiner. He is known as father of blood grouping. Carl Landsteiner. He used to perform the process of blood transfusion among different human, from human to animals, animals to human. And his observation was, most of the time they used to die, his patients. Okay. Now he started finding the bloods are red in color for every human being, all human being. So why it's happening that when I'm trying to transfuse blood from one person to another person, it's causing death and coagulation inside the body, right? Then he started researching on RBC, and he found some glycoproteins. He found some glycoproteins upon the surface of RBC, and he named them antigens. And based upon the presence of those antigens on RBC surface, he came with a new system, okay, that is blood grouping system, and his blood grouping system is known as ABO system. And later on, he and his colleagues also discovered another antigen which are responsible for giving positive or negative blood grouping. It is Rh antigen. So whenever we'll be talking about blood grouping, the determining factor of blood groups, where you can find them into the plasma? No, just on the surface of RBC, and we call those determining factors as antigen, and their biomolecular nature, they are glycoprotein. They are made up of protein and carbohydrate. Okay. So as you know, blood of human being differ in certain aspects, though it appears to be similar. As I was saying that blood is red for every all, right? So various types of grouping of blood has been done after Landsteiner. There are many scientists, they are researching, still researching, and not only we have that ABO system of blood grouping, we have different like other systems of blood groupings are also present. Two such grouping like ABO system and Rh system are widely used all over the world, right? So these two are the widely used blood grouping system, ABO system and Rh system. There are other blood groupings, right? HNRA system, INRA system, golden blood. There are different types of Bombay typing. There are different other types of bloods are also present. So we'll be talking about this ABO system and RH system. Okay. So based upon the presence of the antigens, there are 4 types of blood groups: blood group A, blood group B, blood group AB, and blood group O. Okay. Now, if you check the antigen present on the RBC, the person who is having blood group A, the antigen will be A antigen. The person who has blood group B on RBC will be finding B antigen. The person who has the blood group of AB is a case of codominance. Both A and B antigens are present on the surface of RBC. And the person whom you are calling that the blood group is O, you won't be finding either there is no A antigen or B antigen. There is no A or B antigen present on the surface RBC of the person who is having O blood group. Okay. But you will tell me that, sir, in some books and from my teachers, we got to know that there are certain other determining factors. They are the antibodies. So what about them? Antibodies. So basically, antibodies, they are present in plasma in response to the presence of the antigens into the RBC. So while your body is developing and blood is developing, first there will be formation of antigen on RBC, and after that, okay, for a newborn child, within few days, antibodies will be produced inside the plasma of his or her blood. So first antigen formation is there on the surface of RBC, and as a reaction of that antigen, we can call it adaptive immunity. As a result of adaptive immunity, every human being will be getting some antibodies present into the plasma. So plasma, that's why antibodies in plasma are not determining factor of blood groups, right? So what about the plasma, presence of plasma? If a person is having blood group A, in his or her plasma, you will be getting anti-B antibody. The person having blood group B, in his or her plasma, you will be getting anti-A antibody. The person with blood group AB, who has A and B both the antigen, you won't be finding any antibodies into the plasma. And the person with O blood group, who doesn't have any of the antigens, neither A nor B, both the antibodies anti-A and anti-B are present into the plasma, right? So remember, it's the antibodies who are attacking the antigens. It's the antibodies, they are the villains here. So if the person is having blood group A, the antigen is A on RBC. So that's why there is no presence of anti-B into the blood of that person. If there is presence of anti-B, there is no issue for that particular individual, right? The person with blood B having antigen B on RBC, and in plasma, antibody you are getting is not anti-B, it's anti-A. So there will be no incompatibility between the antigen and antibody in that particular person. Now what about blood transfusion? In blood transfusion, if I am having blood group A and you are donating me blood, you are having blood group B, and if you are donating me blood, then what will happen? Your blood has anti-A, your blood has anti-A, right? And my blood has anti-B. So what will happen? Your blood will attack the antigen of my RBC, and my blood will attack the antigen of your blood. So the blood will be coagulated inside the blood vessel. There will be destruction. That's why if someone asks you, a person with blood group can donate blood to which all groups? He or she can donate blood. He or she can donate blood. I have to make a separate column here. So if this person is donating blood, who are gonna receive, or he or she can donate blood to a person with blood group A, to a person with blood group AB. The person with B blood group can donate blood to a person with B blood group, similar blood group, as well as AB blood group. a person with AB blood group can donate blood to a person who is having AB blood group. A person with O blood group can donate blood group to the person who is having O, who is having A blood group, who is having B blood group, who is having AB blood group. So that's why you can observe that this is the blood group AB. The person with blood group AB can take blood from every person. That's why we call AB blood group as universal recipient. Okay. Now, who can donate blood group to A? A person with blood group A and another person with blood group O can donate blood group to the person A. Who can donate blood group to the person who is having B blood group? A person with B blood group, a person with O blood group. Who all can donate blood to the person with AB blood group? A person with AB blood group, person with A blood group, person with B blood group, person with O blood group. Who all can donate blood to O blood group person? Only the person who is having O blood group. So that's why O can donate to each and every one. So that's why O is considered as universal donor. Now, why it's so? See, the person who is having O blood group, they have no antigen present into the RBC on the RBC surface, but have both the antibodies, antibody A and antibody B present in the plasma. Now, which is villain? Antibodies are the villain. So what happens here? If the person with blood group O is donating blood to a person with blood group A, now who is donor here? The person O. Who is recipient here? The person A. Now remember, always the antibodies present in plasma will try to attack, try to find the antigens from the donor's blood. Now for the person with blood group A, he or she has antibody B into the blood. Now that antibody B will try to find which antigen? B antigen. But for the person blood group O, it has none of the antigens, neither A nor B. So it won't be finding any of the antigens, so there will be no class among them. If that same person with blood group O is donating blood to a person B, now B is recipient, O is donor. Now the B has anti-A into the plasma. Now the anti-A will try to find antigen A. Again, O has none of the antigens, so it will be safe. If the person O is donating blood to a person having blood group AB, the person has no or none of the antibodies present into the plasma. So there will be no clash with the blood, and obviously O can donate blood to person with O blood group. So remember that whenever there is blood transfusion, it's always the antibody of plasma of recipient is attacking the antigen of the donor. Okay. Now if you ask that can the antibodies of donor can attack the antigen of the recipient? In a very mild way. Okay, the antibodies which are present into the donor's blood, they can attack the antigen of the recipient, but that won't make a much difference. Okay, so remember. Now what about AB? Why AB can receive blood from every other blood group? Now let's discuss that. Now suppose A is donating blood to the blood group of, if the person is having blood group AB. Now AB will be the recipient, A is donor. Now AB has no antibodies to attack the antigen. So as because the AB person, they don't have any antibodies into the plasma, so whoever donating them blood, the donor's blood is threat free from recipient's blood if the recipient is AB. So again, I am making it more simple. This is RBC, this is another RBC, this is another RBC. If this is A antigen, only A antigen is present on the surface of the RBC, then we will be calling that person is having A blood group. If this is B antigen, is present on the surface of RBC, then we will be calling that the person has B blood group. Okay. Another third one RBC. If you observe, he or she has both A antigen and B antigen. Then the blood group of that person will be AB, that is a case of incomplete dominance. Okay, now let's talk about another RBC. Now this particular person do not have any of the antigen in between A antigen or B antigen. So that's because A and B both are absent here. So blood group will be calling that person is having O blood group, right? Okay. Now let's move forward. Now if the person who is having A antigen on the RBC, in the plasma, the person will have anti B. The person who has B antigen on the RBC surface, he or she has anti A into the plasma. The person who has both the antigen, the plasma will be nil from any of the antibodies. If the person is having none of the antigen, the plasma will have both the anti-A antibody and anti-B antibodies. So this is outside the RBC, that means this part is the part of plasma. This part is the part of plasma. And remember that when the development of blood group is happening into your body, first there will be formation of antigens on the surface of RBC. After few days, there will be formation of antibodies into the plasma. Okay. Now that was all about the ABO system. Now let's talk about the RH system. Okay. So another antigen, the RH antigen, similar to one present in the rhesus monkeys. So when they were searching for antigen on the surface of RBC, if suppose a person is having B blood group, having B antigen, along with if the person has another antigen on the RBC surface called RH antigen, then we call that person is B positive. If RH antigen is absent on the surface of RBC, then we call that person is B negative, right? So after discovery of these antigens A and B, later on Landsteiner with his colleagues, he found another antigen called RH antigen, and it was for the first time discovered into monkeys, that is rhesus monkeys. So that's why from the word rhesus it got the name RH, right? But a similar, not this particular antigen which you will be finding on the RBC surface of rhesus monkey, a similar kind of antigen you will be finding into the surface of RBC of human being. Okay, so we have kept the name same, RH antigen is also observed on the surface of RBCs of majority, nearly 80% of humans of the world. Okay, such individuals are called RH positive, and those in whom this antigen is absent, they are called RH negative person. And RH negative person, if exposed to RH positive blood, will form specific antibodies against the RH antigen. So thing is that if you are donating blood of RH positive person to the body of RH negative person, there will be immune reaction, because the body of RH negative person never seen this antigen RH positive. So that person will generate RH antibody, and this RH antibody will attack the antigen present on the RBC, and the RBC will be destroyed. So that's why this process is not recommended. You cannot transfuse blood between you cannot donate blood of a RH positive person to a person who has RH negative. But if you are doing the opposite, if you are donating blood of RH negative person to a person who is having Rh positive blood, then there will be no issue. Happily, the person will welcome the blood of Rh negative, right? Why so? Because the person with Rh positive antigen doesn't have any antibodies, and if it's getting Rh negative blood, that blood also don't have any kind of new antigen. That's why you are calling it Rh negative. So if there is no new antigen into your body, or your immune system are not finding any new antigen, then there will be no immune attack against that. So that's why Rh negative person can donate blood to Rh positive person, but Rh positive person cannot donate blood to Rh negative person. So that's why we say that for blood groups AB, AB, O, if the person with blood group A have the Rh antigen, we call it A positive. If Rh antigen is absent, that is A negative. Person with B group having Rh antigen on RBC, B positive, Rh is absent on the surface of RBC, then we call it B negative. Person with AB blood group have the Rh antigen on RBC, AB positive, no Rh on RBC, AB negative. A person with O blood group, if Rh antigen is present on RBC surface, O positive. If Rh antigen is absent, that is O negative. And as because the negative person can donate blood to positive person, so who is the universal donor? O blood group. Person with O blood group, they are universal donor. In true manner, O negative blood group persons, they are called true universal donor. So they can donate blood to all the person with negative blood group and to all the person with positive blood group. And which person is considered as true universal recipient? Positive. The person with positive blood can receive the blood of negative. So AB positive, a person with AB positive blood called as universal, true universal recipient, they can receive blood from all the positive blood group, all the negative blood group. Okay. Now let's solve a question related to that. Which of the following statements is incorrect? You have to find the incorrect statement. It's saying that a person of O blood group has anti-A and anti-B antibodies in his blood plasma. So this is true statement. A person of B blood group cannot donate blood to a person of A blood group. This is also true statement. Option 3 said that blood group is designated on the basis of the presence of antibodies in the blood plasma. Already we were discussing this question, that blood group is designated not on the basis of presence of antibodies in plasma, but on the presence of antigens on RBC. Because first there will be formation of antigen on RBC, and after that, after few time, in some detailed lecture, I will be drilling this particular concept. After few time, the body of that person will develop antibodies into the plasma. Fourth option say that the person of AB blood group is universal recipient. So the incorrect option is option number 3. Okay. Now related to the blood group, we need to study a very important disease that is called hemolytic disease in newborn, or it's called also, we call this hemolytic disease in newborn as erythroblastosis foetalis. Now it's a very conceptual topic to understand. Listen very carefully. What is hemolytic disease in newborn? So in fetus, what happening basically, there is antibody attack on the RBC, on the liver. So that's why there will be destruction of RBCs happening inside the body. So that's why we are calling it hemolysis, and that is observed into fetus or newborn babies. So that's why hemolytic disease and newborn. Now why it's called erythroblastosis foetalis? Is because observed in fetus. And why it's erythroblastosis? When the normal RBCs or mature RBCs are getting attacked, so they are getting destroyed. So in fetal blood stream, you cannot find mature RBC, only the remaining immature RBC that we call erythroblast. Remember the process of erythropoiesis? Only the erythroblasts are remaining into the fetus blood stream. So that's why you will be finding only immature RBC. That's why erythroblastosis foetalis, because mature RBC they are getting destroyed. Now why it's happening and how it's happening? So suppose there is a marriage between father with Rh positive blood and a mother having Rh negative blood. As because this Rh is a dominant allele, the first child will carry the Rh antigen, so it will be Rh positive, right? The first child will be carrying the dominant allele from the father. So there is this is not an issue. Now what will happen during the time of the childbirth of this or delivery or the parturition of this first child, through the placental barrier, through umbilical cord and placenta, is because rupture will be there, the blood from that first child, blood from the first child will enter into mother's bloodstream, and the mother is Rh negative, and the blood from the first child is getting inside the mother, that is Rh positive. So what after that, during the time of the delivery of the first child, mother, till now the mother blood was unknown about this Rh antigen, but as soon as the blood is exposed, the mother blood will start formation of Rh antibodies as immune response. Now the mother blood, after the delivery of the first child, the mother blood has Rh antibodies which are IgG in nature. These Rh antibodies, they are IgG in nature, the smallest of all the antibodies, IgG. Now what about during the time of second pregnancy? Now during the time of second pregnancy, when it's turn of second child, the mother blood which has produced Rh antibodies which are IgG in nature, now that can cross the placental barrier, that will cross placental barrier and will get inside the bloodstream of second child and will attack RBC, will attack liver, and upon destruction of mature RBC and liver, that fetus will be suffering from anemia, severe anemia and jaundice, right? And you will be finding only immature RBCs inside the bloodstream of the fetus. That's why we are naming that. Because you are finding only erythroblast. Immature RBC. Erythroblast doesn't mean the erythrocytes are getting blast. Immature RBC, presence of immature RBC, because mature RBCs are destroyed by the Rh antibodies. So this condition we call it erythroblastosis fatalis, but there is a prevention methodology. Now let's look at that. So this can be avoided by administration of anti-Rh antibodies. Anti-Rh antibodies and Rh antibodies are not similar. Now to prevent this erythroblastosis fatalis, just after the birth of first child, doctors can inject antidote called Rogam, basically that contains the anti-Rh antibodies which will kill or neutralize the Rh antigens just after the first or initial exposure, and to the mother immediately after the delivery of the first child. NCERT is saying just immediately after the delivery of the first child, you need to inject that Rogam antidote or anti-Rh antibodies. So that was all about erythroblastosis fatalis. Now let's solve few questions. The question is saying that adult human RBCs are enucleated. Which of the following statement is or most appropriate explanation for this feature? Okay, they do not need to reproduce. It's not like that. They are somatic cells. No relevancy. They do not metabolize. No. When after enucleation, RBC will be performing metabolism, but the rate will be slower. So the main primary reason will be their internal space is available for oxygen transport. You know that 35% volume of RBC will be occupied by HB, means hemoglobin. Yes, so we got our answer option 4. So option 1, that is 4 only, that is the relevant option why RBCs are enucleated. Now jumping to the next topic, that is coagulation of blood or blood clotting. Okay, so basically coagulation, after a damage or injury into our tissue, the process is initiated, right? The heparin is getting inactivated and few enzymes are responsible for that. Not only one single enzyme, there are 13 clotting factors, and that will be initiated by the damaged tissue. The clotting process will be initiated by the damaged tissue and some platelets, right? So that's why it's written here, coagulation of blood, that is a series of enzymatic reaction or cascade. Cascade means one enzyme is activating another enzyme. That's why we are calling this a cascade phenomena of blood coagulation. Okay. Now what happens during the time of blood clotting? Why you need to clot the blood? When there is a damage or rupture of blood vessel, the blood will be oozing out or coming out along with the RBC and other components, right? Okay. Now this damaged place, the damaged region, assume that the cells will start crying, they will secrete some important factors, right? And those factors will invite more and more number of platelets towards the ruptured blood vessel or damaged tissue. And as soon as the platelets will be arriving, they will initiate the process, and through the completion of a long cascade, there will be formation of fibrin protein, which is already present inside the blood but in inactivated form, that is fibrinogen, the largest clotting factor of the plasma protein that is present in fibrinogen. So the motto is to activate the fibrinogen. So what is the motto? To activate fibrinogen. When it's getting activated, it will produce fibrin, and they are fibrous in nature. No more they are soluble or dissolved. Now they will become insoluble, and this fibrin protein will form a network or a mesh, and that mesh or network will keep trapping the RBCs, and these trapped RBCs will be glued by gelatinous substance. The trapped RBCs will be glued by a network of gelatinous substance and fibrin, and that plaque, that clump, we call it clot, blood clot or thrombus. Right, now let's study the process of coagulation of blood. So when there is tissue injury, tissue will release tissue thromboplastin. Also we call it exothromboplastin. Now blood vessels are also injured, and so platelets are also called. The platelets will release platelets thromboplastin or platelets factor. Also we call it endothromboplastin. So the thromboplastin which is produced by tissue, we call it exothromboplastin. The thromboplastin which is secreted by platelets where the blood vessels are ruptured, it is called exothromboplastin or platelets factor. Now for the activity, here we need another clotting factor, that is calcium. Without calcium, blood clotting is not possible. Now with the help of this calcium, exo and endothromboplastin will catalyze a new reaction. And what kind of reaction will be done? They will form an enzyme, thrombokinase, right? Now once the thrombokinase is produced or thrombokinase is formed, that will activate the inactivated protein or enzyme prothrombin, and the activated one is thrombin. Okay, so prothrombin is getting converted into thrombin by the enzyme called thrombokinase or prothrombinase, and again this thrombin will act as an enzyme that will catalyze one more reaction. Now in blood we got the inactivated fibrin, that is fibrinogen. They're dissolved into the plasma. So as soon as thrombin is produced, the fibrinogen is getting converted into fibrin. Now the fibrin will form mesh of fibers, and that mesh of fiber will trap RBCs, and eventually they will form the clot. So this is how the process of blood clotting is done, and here you must name another important component of our body, or nutrient, that is vitamin K, which is necessary, vitamin K, which is necessary for the formation of prothrombin. Though it's not directly involved, but you need vitamin K in your body for rapid up the production of prothrombin, right? Now let's solve this particular question. So the question does ask the a reduction in number of platelets. If the number of platelets are reduced into your body, see, when you are suffering from dengue, then doctor will be telling you, just give me the platelet count of that patient, right? So in some disease, there is chance that platelets, number of platelets are deteriorating into your body, and you need to measure that. Why? Will they produce blood coagulation inside the blood vessel? No. Will they promote clotting disorders? Yes. Excessive loss of blood from the body, if there is less platelet, obviously coagulation will not be there, excessive loss of blood will be there. So we got our answer, that is option 4, both B and C. Okay. Now let's dive deep into another system. Right now we were talking about the prime body fluid, that is blood. Now let's talk about lymph. The lymph is also called the filtered blood or the tissue fluid or the middleman of our body. Now why it's so? See, blood is a part of closed circulation into our body. Heart is pumping the blood and blood is circulating through the blood vessels of different diameters, right? But blood hasn't got the direct exposure to the cells of our body, right? Directly. Blood is not directly in contact with the cells. So that's why blood needs an assistant here, and that's why few part of the blood will be getting filtered out from a special kind of capillaries, blood capillaries, and that filtered blood will go inside the tissue, and that will make a bridge between blood and the cells of a tissue. That's why we are calling it tissue fluid, or a middleman that is acting as a middleman in between blood and the tissue, blood and our body cells, right? So let's see what NCERT is saying. As the blood passes through the capillaries in tissues, some water along with many small water soluble substances move out into the space between the cells of tissue, that is called intracellular space or interstitial space. The larger proteins and most of the formed elements in the blood vessels. Okay. Again, try to understand. When the blood is flowing through a particular tissue, some parts of the blood will be getting filtered out with the help of some particular capillaries. And what are the components will be filtering out, and what are the components which will be retaining? Many small water soluble substances move out into the tissue space or interstitial space, along with few formed elements, mostly the lymphocytes, the T cells and B cells. What are the things which will not be coming out? Larger proteins will not be coming out, and most of the formed elements, like most of the WBCs, right, and RBCs will not be coming out. So the lymph has all the minerals of plasma, few proteins of plasma, smaller proteins, few WBCs like lymphocytes, and few platelets. Okay. So the fluid released out is called interstitial fluid, because the tissue space is also called the interstitial space, or the fluid present inside that interstitial fluid or tissue fluid. It has the same, now the important line is here, it has the same mineralic distribution as that of plasma. It has the same mineralic distribution as that of plasma, because the concentration is more or less similar to blood and plasma, and there is near about 300 milliosmol per liter volume. That is the concentration of blood as well as plasma. Why concentration is similar? Because the minerals and the water components are similar in plasma of blood and in lymph. Okay. Exchange of nutrients, gases between the blood and the cells always occur through this fluid. That's why we are calling it middleman. Lymph is a colorless fluid because there is no RBCs are present, containing specialized lymphocytes, rich in lymphocytes. That's why the name of the fluid is lymph, which are responsible. Now let's talk about their function for the immune responses of tissue. Is also an important carrier of nutrient and hormones. Fats are absorbed through the lymph in the lacteals present in the intestinal villi. So when we talk about the digestion and absorption process, the digested carbohydrates, the digested proteins are directly absorbed by the blood capillaries, but because the fats or the digested fats are present into large globular format, blood capillaries are unable to absorb them. So there we need the lymph capillaries, or we call them lacteals, as because they are milky in color. That's why you are naming them lacteals. They are responsible for the absorption of fats from the small intestine, right? So if someone asks you what are the function of lymph, getting nutrients from the blood and delivering to the tissue, getting oxygen from the blood and delivering to the tissue, getting carbon dioxide and excreta from the tissue and delivering to the blood. Also, they are responsible for providing immunity to the tissue and cells, as because they contain T cells and B cells, right? So now let's clarify that thing. So this is a blood stream. These are the tissue cells. And this is the interstitial space. So special kind of capillaries you will be finding into some tissue. They have tiny pores into them. We call them fenestrated capillaries. So with the help of these tiny pores or the fenestra, few part of the blood will get filtered out into the tissue space or interstitial space and will form a colorless fluid with some T cells and B cells. That fluid is called lymph, where this lymph is produced into the tissue space, which we call interstitial space, right? Now, the lymphs will be collected by lymph capillaries, and the lymph capillaries will join together to form lymph vessels. From lymph vessels, you will observe formation of lymph ducts, and in many parts of the lymph ducts, you will observe some swelled parts that are called lymph nodes, and ultimately they will deliver the expired lymph into the venous blood. So lymph is produced into the tissue space, and the expired lymph for the recycling process will be released into the venous blood into our body, right? So that is how your lymphatic system works. And as because the lymph is produced into open space, you can consider that as a open circulation. The lymphatic system is a kind of open circulation, and our cardiovascular system is closed circulation, right? Now let's solve one more question here. Which of the following is correct? You have to choose the correct answer. So lymph is plasma plus RBC plus WBC. No. Blood is plasma plus RBC plus WBC plus platelets. Yes, this is our correct answer. plasma equal to blood minus lymphocyte? No, this is incorrect one. Serum equals to blood plus fibrinogen? That's also wrong option. So this is our correct answer. I'm recapping one more thing. So what is lymph? Lymph is plasma minus RBC and large proteins. If you ask me that will there be observation of clotting inside the lymph? Yes, lymph can be clotted, but it will take more time in compared to blood. Okay, what is serum? Serum means plasma minus clotting factors. That is serum. If you delete the clotting factors from plasma, then the colorless fluid we will be getting, that we will be calling it serum. Okay. What is plasma? Plasma means blood minus formed elements. Blood minus RBC, WBC platelets, that straw colored fluid, we call it plasma. Okay. Now again a new topic we are going to cover here, there is circulatory pathways. So we need to talk about the open circulation and closed circulation. So if you observe the undeveloped of the ancient animals, open circulation is predominant, but in advanced one you can observe closed circulation. Okay. Now what is open circulation basically? Open circulatory system. In open circulatory system, the blood is getting circulated from heart to body cavity, which we call sinus, and again from body cavity to heart. So the blood is not confined within the blood vessels. That's why that is called open circulation or open circulatory system. You can observe them into arthropoda and mollusca, specifically the gastropods, right? Okay, there are few mollusca, they will exhibit closed circulation, but most of the molluscans will be finding open circulation into them. Okay, as well as there are few chordates also, they exhibit open circulation, they are called urochordata. Okay, so if we check the circulation pattern inside the body of cockroach, then you observe that the dorsal side there will be heart. The heart is present on the dorsal side. The heart is pumping the colorless blood into the body cavity or sinus. The blood is flowing throughout the body and again taken back into the heart and pump the heart, the chamber of hearts, they are pumping the received blood to the chamber frontwards. So this is how there is continuous circulation of blood in between heart and the body cavity or sinus. Okay. Now let's talk about the closed circulatory system. Closed circulatory system, which is observed in annelids like earthworms and in chordates. Okay. So in closed circulation, what is the perfect definition of closed circulation? Blood is confined with vessels and flowing through vessels of different diameters. Okay. Presence of heart is not mandatory for a closed circulatory pathway. Okay. Heart, though most of the animals which exhibit closed circulation, they have heart, but there are few animals, those who have closed circulation, but they don't have heart. So you need to provide a pathway. You need to provide a pathway made up of blood vessels of different diameters through which the blood will flow from one place to another place. So this simple system can be called as closed circulation, as because the blood is not coming direct contact with the body tissue or cells. So when the blood is getting circulated between heart to blood vessel and again blood vessel to heart, or when the blood is getting circulated within a closed network of blood vessels only. That is observed in Amphioxus. They don't have heart, but they exhibit closed circulation. Okay. Now, what is the advancement, the importance of closed circulatory system over open circulatory system? It is written here, flow of fluid can be more precisely regulated. So, basically, in open circulation, the materials are evenly distributed among the body cells. But what we know that different tissue or different cells, they have different requirement for energy, for nutrients or for oxygen. So that's why if you want to provide a particular tissue more nutrients and another tissue less nutrients. So specifically, if you are becoming more specific towards the delivery of the substances, then you need closed circulatory system. Open circulatory system won't be working in that case. Right. Now let's talk about the evolution of heart in vertebrates. Right. Okay. So what you can observe that the primitive two-chambered heart is observed into fishes, and the two-chambered heart is called venous heart, where you can observe single circulation. Now, what do you mean that? So, this is a venous heart. This is the venous heart where there is one auricle and one ventricle. So, that's why we are calling it two-chambered heart. So, the auricle is getting deoxygenated blood. Auricle is getting deoxygenated blood, carbon dioxide rich blood through a chamber called sinus venosus, and that auricle is pumping that deoxygenated blood to ventricle, and ventricle is pumping that deoxygenated blood towards the body, right? So, what's happening? A heart which is receiving deoxygenated blood and pumping deoxygenated blood, that heart is basically acting as a vein. So, that's why you are calling as venous heart or two-chambered heart. And the circulation is single circulation because the blood is passing the heart once and the blood is deoxygenated in nature. Now, what about the amphibians and reptiles? They have three-chambered heart, though reptiles have 3.5 chambered heart, but we will be considering three-chambered only. That's because for their heart, incomplete ventricular septum is formed. So that's why if the ventricle formation, the chamberization of ventricle is incomplete, so we will be considering there is one single ventricle and two auricles, right? So in case of heart of amphibians and reptiles, they have two auricles and one ventricle. So that's why they have three-chambered heart, and the circulation mode is mixed double circulation or incomplete double circulation. Now, what do you mean that? Both the auricles, they are getting separated blood. The right auricle getting blood with carbon dioxide and the left auricle getting blood with oxygen. So, oxygenated and deoxygenated blood is separated into auricular level or atrial level, but when the blood is coming from both the auricles into the ventricle, ultimately it is getting mixed, and ultimately ventricle is the chamber which is responsible for pumping out the blood towards the body organs. So, the ventricle is pumping the mixed blood, which is basically becoming impure. That's why we call that mixed double circulation or incomplete double circulation, is because though they have two separate auricles, but one single or incompletely separated ventricles. What about mammals and birds? That the advanced animals of vertebrates, they have four-chambered heart, and see, for reptiles there is one exception, that is crocodile. So mammals, birds and crocodile. Crocodiles also joining the party. Mammals, birds and crocodiles, they have four-chambered heart, and the process or the mechanism of circulation, we call it double circulation, is because the left side of the heart is receiving and pumping oxygenated blood, right side of the heart is receiving and pumping deoxygenated blood. Heart is completely chamberized into 4 chambers, 2 auricles and 2 ventricles. That's why this mechanism is called double circulation. Successfully, the heart can separate out oxygenated blood and deoxygenated blood. Okay. Now, we will be moving towards our circulatory system. Though we have finished the discussion of blood, now we are moving towards the main blood pumping station of our body, that is heart, right? So, we have a muscular heart that's called myogenic in nature, is because the heartbeat is produced by its own cardiac muscles. That's why we are calling autorhythmic heart or myogenic heart. Okay. Our circulatory system also have some blood vessels with different diameters, arteries, veins, and the thinnest one, they are capillaries, right? The thick-walled blood vessels, arteries, thin-walled blood vessels, veins, and the ultra-thin-walled blood vessels, they are capillaries. Okay. Blood, already we have spoken about blood, and all these components are mesodermally originated. Now let's talk about the location of our heart. So we have one pair of lungs. Right lung and left lung. So here you can observe there is a concave surface present in the left lung, that is called cardiac notch. That's called cardiac notch. And the space in between the lungs, we call it mediastinum. So into that mediastinum, the apex of heart, this conus part of heart, the conic part of heart, tilted towards the left. This is the location of heart, the cardiac notch of the left lung, and obviously that is present into the thoracic cavity. Our heart is not present to the left side, our heart is tilted towards left. The apex of heart, this part of heart is called apex. This part of our heart, this is called base. And the size of our heart is like our clenched fist. This is the clenched fist. So our heart is tilted towards the left, occupying some volume of our left lung, that is cardiac notch. In between the space of left and right lung, that is mediastinum of thoracic cavity. Okay. Now I will be talking about the morphology and anatomy of heart. If this is our heart, it's always contracting and relaxing. That is called systole and diastolic movement is performed. So that's why we need to protect our heart from any kind of shock or any kind of friction. So the protector covering of heart is called pericardium. Okay. The outermost pericardium, there is fibrous layer, there is fibrous pericardium. If you peel off this pericardium, you will be getting another pericardial layer, that is called serous pericardium. And again there are two layers in the serous pericardium, the outer serous pericardium and the inner serous pericardium. So ultimately, if someone asks you what is the covering of heart, then you need to answer it's pericardium. Right. Now, what about the layers of heart? The outermost layer of our heart is called epicardium. The middle but muscular layer of heart, that is called myocardium. And the innermost layer, which is made up of tessellated or simple squamous epithelium, that we call it endocardium. Wall of heart. Right. Now let's talk about the chambers. Heart is divided into 4 chambers. Okay. So the upper chambers which are responsible for receiving the blood, the lower chambers which are responsible for the pumping the blood. So the upper chambers are called atrium. This is the left atrium, LA. This is the right atrium, RA. So right atrium and left atrium. Ventricle, 2 ventricles, left ventricle and right ventricle. 2 ventricles and 2 atria, right. Now we need to separate the chambers from each other. Now we will be separating. For that we need wall, and that muscular wall which is separating the right and left side, that is called septum. Now a very important thing I am telling, the septum which is separating the left and right ventricle, that is called intraventricular septum. The septum which is separating the right and left atrium, that is called intraatrial septum. Both the intraatrial and intraventricular septum, they are muscular in nature. Intraatrial septum and intraventricular septum, they are muscular in nature. In between them, these 2 septum are separated by a fibrous connective tissue septum, that is called atrioventricular septum, right. Okay. Now which are the components separating the upper chambers and lower chambers? See, left atrium and left ventricle, they are separated by a valve called bicuspid valve or mitral valve. Right atrium and right ventricle separated by another valve called tricuspid valve. Now why bicuspid and tricuspid? I am explaining it. Here it is. Separator of right atrium to right ventricle. It has 3 cusps. A valve having 3 cusps. That's why we are naming it tricuspid valve. Tricuspid valve having 3 cusps. 1, 2, 3. Okay. The separator of left atrium and left ventricle having only 2 cusps, that also looks like the cap of the fathers of the church, that is called mitre. Look like a cap of father, that's why it's called mitral valve, right. Their fibrous structure. We'll be talking about the plumbing system, like what are the arteries and veins which are originating or linked with every chamber. Okay, so if we talk about the left atrium, 4 openings, you'll be getting 4 openings coming from 4 veins coming from your lungs carrying oxygenated blood. We call them pulmonary veins. Four pulmonary veins. Okay, they are delivering blood from lungs to left atrium, right. Now coming to right atrium, okay, the principal veins of our body, the main veins of our body which are carrying deoxygenated blood. See, the pulmonary veins, they are carrying oxygenated blood, but there will be two important veins, that is superior vena cava coming from upside and coming from downside, inferior vena cava. We also call them systemic veins. So these two prime systemic veins, they are getting opened or releasing deoxygenated blood collecting from all the organs of your body into the right atrium. Right atrium also has another opening of coronary sinus. So right atrium has three openings, one opening from superior vena cava, another opening from inferior vena cava, and another opening of coronary sinus, which is present just near to the back to the right atrium. Okay, now let's come to the ventricle. So both the atria, they are the receiving chamber. Left atrium is receiving oxygenated blood, right atrium receiving deoxygenated blood. Now the oxygenated blood will be pumped from left ventricle and the deoxygenated blood will be pumped from right ventricle. Okay. Now how deoxygenated blood is getting pumped from right ventricle? So for that you have this T-shaped artery called pulmonary artery. An artery which is carrying deoxygenated blood. Okay. Now from the left ventricle, the artery, the J-shaped or the C-shaped artery, which is transporting oxygenated blood to the organs and tissue of your body, that is called systemic artery or aorta. And is carrying oxygenated blood. Right. Now you will be getting some flaps, some luna, half moon like flaps in the opening of the pulmonary artery as well as the opening of aorta or systemic artery. So these moon like flaps, we call them semilunar valves. We call them semilunar valves. So the semilunar valves at the opening of pulmonary artery, they are called pulmonary semilunar valves. They are 3 in number. And at the opening of aorta, systemic artery, the semilunar valves, we call them systemic semilunar valves or aortic semilunar valve. Aortic semilunar valves. Here also there are 3 in number. They look like semi plus luna. Semi lunar. Luna means moon. Yeah. So half moon shaped. They are half moon shaped valves. That's why we are calling them semilunar valves. They are girding the 2 major arteries, that is pulmonary artery and systemic artery, to prevent the backflow of blood from these arteries inside the ventricles. Right. Tricuspid and bicuspid valve, they are preventing backflow of the blood from ventricles to atrium. Right. Now there is a tiny ligament you can observe in between the pulmonary artery and the aorta, that is called ligamentum arteriosum. This is a remnant structure. Okay. Now on the wall of the aortic septum, intra-atrial septum, you will get an impression called fossa ovalis. A tiny impression present on the right side of the intra-atrial septum, that is called fossa ovalis. There is the remaining of a foramen ovalis. During the time of our embryonic development, actually at first the right atrium and the left atrium, they were connected through an opening called foramen ovalis. Now upon maturation, the foramen ovalis will get filled and the impression will be present in the form of fossa ovalis on the right side of the intra-atrial septum. Okay, so this is all about the structure or the anatomical structure of human heart. So this part, what we call, remember, that is the apex. Okay, now let's elaborate a bit about the structure of our heart. So if we talk about the valves, so what are the different valves? Bicuspid valves, they have 2 cusp. Tricuspid valves, they have 3 cusps. We have semilunar valves into our heart. Okay, so out of the 4 chambers, right atrium, left atrium, right ventricle, left ventricle, the largest chamber is left ventricle. Right atrium is separated by left atrium by interatrial septum, which is muscular in nature. Right ventricle is separated from the left ventricle by interventricular septum, again which is muscular in nature, and the septum which is separating the atriums and ventricles in the middle, that is called atrioventricular septum, that is fibrous in nature. Okay, now we'll talk about the conducting system of human heart, just because we know that heart is continuously showing systole and diastolic movement, and from where, if you ask that from where the heart is producing such energy and force, okay, so heart is producing its beat or systolic and diastolic force by the help of specialized cardiac muscle, we call it nodal tissue. So it's written here, a patch of tissue is present in the right upper corner is called the sinoatrial node or SAN. So a patch of tissue which is present at the right upper corner of the right atrium, this is called SA node, a sinoatrial node or SAN. That is the mega producer or the generator of heart that can produce 70 to 75 depolarization or heartbeat in a minute. Okay, another mass of this tissue is seen in the lower left corner of the right atriums. The lower left corner of the right atrium, another mass of the tissue is present, that is called AV node or atrioventricular node. AV node or atrioventricular node. So one is SAN or sinoatrial node, one is AVN or atrioventricular node. They are present in the right atrium, one or the upper right corner, and the AVN is the lower left corner, and they are connected by internodal fibers. Okay, what after that? AV bundle or bundle of His passes through the atrioventricular septum. Now what after AV node? After AV node, a bundle is produced, that is called AV bundle or bundle of His, that will get inside through the atrioventricular septum, and when enter into the ventricular wall or ventricular septum, that will get bifurcated into the right bundle of His and left bundle of His. And remember, the left bundle of His is thicker than the right bundle of His, because the left side, the left ventricle, is the strongest chamber of heart. Remember this sentence from the Spider-Man: with great power comes great responsibility. Yes, the example is left ventricle. Why it's largest chamber of heart? Why it's strongest chamber of heart? Because Almighty has given the most important responsibility, that is to pump oxygenated blood to each and every organ of our body. So that's why the bundle going through it is also thicker. Okay, Purkinje are the present throughout the ventricular musculature. Now upon entering into the ventricular muscles, this bundle of His will get branched. The branch of bundle of his, left and right bundle of his, we call them Purkinje fiber. So the nodal tissue work in this manner. The generator, the pulse will be started by SAN, SA node. It will be distributed throughout the musculature of the atrial wall, and then it will be sent to atrioventricular node or AV node, and there will be a little bit delay of flow of those depolarization which is passing through this bundle into the atrioventricular node. This is because when these ions are passing, when the pulse which is generated by SAN is passing through atrioventricular node, they will face little bit difficulty, as because the channels through which the ions will be passed from one cell to another cell, that is through the intercalated disc, through the gap junctions, are narrow into atrioventricular node. So there will be a little bit delay, and after that delay the signal will be transferred into the AV bundle or bundle of his. Now this bundle of his will get bifurcated into left and right bundle, and ultimately they will enter into the ventricular muscle, and they will get divided into numerous branches called Purkinje fibers. So starting from SAN, ending into the Purkinje fiber. If you observe your heart, two upper chambers, two lower chambers. Here your SAN is present, here your AVN is present. From AVN, bundle of his will generate, they will get bifurcated into left bundle and right bundle. This is left bundle, this is right bundle, and again they will get branched as Purkinje fiber, and thus the impulse which is ultimately produced by SAN will be distributed among the cardiac muscles or cardiac musculature which is responsible for our heartbeat. Okay, and it said that SAN generates 70 to 75 depolarization per minute. So our heart rate is average 70 to 75 or 72, is right? So that is due to the production of the beats with the help of SAN. That's why we named SAN as pacemaker. So here pacemaker means formation of the depolarization of the heartbeat, and the AVN is because it's distributing the beats from here throughout the ventricular wall. So AVN is called pacesetter. Okay, now let's solve this question. All vertebrate possess a muscular heart. It should be. Fish, they have two-chambered heart, right? With atrium and ventricles. Amphibians and reptiles have three-chambered heart. Birds and mammals have four-chambered heart. So choose the correct option. So all vertebrate possess a muscular heart. Fish two-chambered, amphibians, reptiles three-chambered. There is one exception in reptiles, that is crocodiles, they have four-chambered heart, and all the birds and mammals, they have also four-chambered heart. Next, well, moving towards the next topic, that is cardiac cycle. Yes, cardiac cycle. The sequential phenomena of our heartbeat. Continuously our heart is performing rhythmic beating starting from our birth to death, right? So that sequential contraction and relaxation is called systole and diastole. Contraction means systole, relaxation means diastole. During the time of systole, heart is pumping blood. During the time of diastole, heart is receiving the blood. Remember this. So cardiac cycle is divided into diastolic phase and systolic phase. Diastole, period of relaxation, systole, period of contraction. And so one cardiac cycle takes 0.8 second. One systole and diastole, one beat takes 0.8 second, right? and that can be further divided into systolic and diastolic phase. So when you talk about this cardiac cycle and their divisions, that is divided into three phases. First starts with joint diastole, then atrial systole, and then ventricular systole. For joint diastole, you need 0.4 second. For atrial systole, you need 0.1 second, the shortest one. For ventricular systole, you need 0.3 second. So 0.1, 0.4, 0.1 and 0.3, it's ultimately 0.8 second. Okay. Now, if the atrial systole is of 0.1 second, the atrial diastolic phase will be for 0.7 second. If the ventricular systole is of 0.3 second, the ventricular diastolic phase will be for 0.5 second. Okay, so that's why if you observe this joint diastole, there you can observe both the ventricle and atria, they are in diastolic phase. So that's why we are naming it joint diastole. Atrial systole consuming 1 second. So apart from this 1 second, everything else, rest 0.7 second is for atrial diastole. Ventricular systole is consuming 0.3 second, rest 0.5 second is for ventricular diastole. And when the atrial diastole and ventricular diastole are colliding with each other, that's forming the joint diastole. Okay, so it's written here, the sequential and repetitive contraction and relaxation of the chambers of our heart is called cardiac cycle. Okay, let's start with systole and diastole. Systole means contraction, and why we need to contract the heart to pump the blood outside, why we need to relax, or when you observe the diastolic phase, when our heart receives the blood. So with proper diagram, I have elaborated them. So during the time of this systole, during the time of diastole, the chambers will welcome the blood. The chambers will welcome the blood. So that's why the emoji is saying welcome. During the time of systole, the heart is pumping the blood. That's why the emoji is saying to the blood, please get out from the chambers. Right. Now let's start with the first phase, joint diastole. The arrow outside means that is diastolic phase. The arrow inside means that is systolic phase. Okay. So at the beginning of the joint diastole, all the four chambers, that is right atrium, left ventricle, right ventricle, they are at diastolic phase, and all the valves are closed, both the bicuspid and tricuspid valves and the sublinar valves, all the valves are closed. But soon after what will happen? The bicuspid and tricuspid valves are open now. So as soon as the bicuspid and tricuspid valves are open, the blood from the atrium, both the atrial chamber from the right atrium and the left atrium will enter into the ventricle, and that is called passive filling of the blood. So the beginning of joint diastole, what we can observe? Bicuspid and tricuspid valves, they are called AV valves, are opened. When AV valves are open, see, all the chambers are relaxing, they are in diastolic mode. So why chambers are going into diastolic mode? They have to receive the blood. So ventricles, they have to receive the blood from atrium. Blood will flow from atria to ventricles. A to V means atria to ventricles, and this process we call it passive filling. Now a question can be asked that what percentage of blood is coming during the time of passive filling? 70% of blood will be flowing from each atrium to one ventricle. Okay. Now if we talk about the left ventricle, then total volume of left ventricle is 120 ml. Okay. Now, while this left ventricle, we are assuming the left ventricle is empty now. Now, empty means it doesn't have the 0 ml blood. It has little amount of blood. What amount of blood it's having? It's having 50 ml blood already. So, if I am telling that my ventricles are emptied, that means still after the prior pumping, we have still 50 ml of blood present inside the left ventricle. Now, what ml or what volume space is left? 120 minus 50, total 70 ml volume is left. So, if I am telling during the time of passive filling, 70% blood is coming from atrium to ventricle. So, what will be the 70% of 70 ml? That is near about 49 ml. So, 49 ml blood is coming from one atrium to one ventricle during the time of joint diastole. So, after joint diastole, another 49 ml blood will come and fill the ventricles. Okay. Now, the second phase, that atrial systole. Now, what will happen during the time of atrial systole? The SAN will generate the impulse. The SAN is switched on now. Before this phase, SAN was switched off. Now, as soon as the SAN start producing the impulse, the atria will go for systole. That's why we are calling it atrial systole. The atria will squeeze the blood present inside forcefully into the remaining part of the ventricles. So, during the time of atrial systole, what happens? SAN produces depolarization, and after that atrial muscles contracted and 30% blood flows from atria to ventricle by the process of active filling. Before this was passive filling. The blood was coming by nature. Now, as because the atrium, they are creating force, squeezing out the blood from atria to ventricle, and 30% blood will come. That is near about, during the time of active filling, near about 21 ml blood will be flowing from one atrium to one ventricle. What I said, the total volume of the left ventricle is 120 ml, right? So, before atrial systole, it was filled with 99 ml. So, how much left is remaining? How much volume is remaining left? 21 ml. So, once the active filling has been done, 21 ml of blood will pump through from one atrium to one ventricle. Now, these ventricles are filled with blood. Okay. Now, the heart will undergo the ventricular systole. We are entering into the third phase of the cycle, that is ventricular systole. Now, at the beginning of ventricular systole, see, the beginning of ventricular systole, what we can observe, the ventricles, they have started contraction, but the atrial chamber, they are relaxing now. Okay. The atria, they are getting blood. They are getting blood. The ventricles are filled with 120 ml of blood. Now, what is the duty of ventricle? The ventricles are undergoing systole to pump the blood inside the pulmonary artery and aorta. Now, the pressure will be created here. Now, there is a chance of backflow of the blood into the atrial chamber. So, to ensure there is no backflow, you have to close the bicuspid and tricuspid valve. So, at the beginning of ventricular systole, we can observe closure of AV valves. That means closure of bicuspid and tricuspid valve, and that produces first heart sound that is lub. So the beginning of ventricular systole, to prevent the backflow of the blood from ventricle to atrium, the bicuspid and tricuspid valves are closed. So this will make a sound called first heart sound lub. Okay. Now upon the ventricular systole, that will create more and more force towards the semilunar valves. Now the semilunar valves are open. Semilunar valves are opened due to the force generated by the ventricular systole, and the blood is pumped into arteries. The blood is pumped into the arteries. From right ventricle, the blood will enter into the pulmonary artery. From left ventricle, the blood will enter into the systemic artery or aorta. So in one stroke or one pump, how much blood is pumped from left ventricle? Left ventricle will be pumping 70 ml of blood. So after the ventricular ejection or ventricular systole, still the ventricle is having 50 ml of blood into it. Okay. Now the heart is preparing, or the ventricle is preparing after pumping. The ventricle will be preparing for what? Ventricular diastole, and the atrium, the left atrium, the right atrium, they're still in diastolic mode. So when there will be starting of ventricular systole, ventricular diastole, atria is already into the diastolic phase, and the new joint diastole, the new cycle gonna start here. But prior to start a new joint diastole or ventricular diastole, what you have to do, you need to close the semilunar valves of the systemic and pulmonary artery. Otherwise, the blood will do a backflow. There is a possibility the blood can flow back from the arteries to the ventricle. To ensure there is no backflow, you need to close the semilunar valves. So at the beginning of ventricular systole or joint diastole, you can observe closure of semilunar valves, and that will create the second heart sound, that is dub. Okay. So what's the heart rate? Heart rate is average 72 per minute. That means our heart is beating 72 times in a minute. What is cardiac cycle? How we can calculate cardiac cycle? In a minute there is 60 seconds and there are 72 beats. So what will be the beating time of heart for one beat? So it will be 0.8 second. So that's the duration of our cardiac cycle. If the heart is beating 72 times in a minute, then for one beat it needs 0.8 second. And when the ventricular systole was going on, ventricle will pump 70 ml blood into the arteries. That is called stroke volume. Okay, left ventricle or right ventricle pumps, each ventricle pumps 70 ml of blood during the time of ventricular systole. That is called stroke volume. And how we can get cardiac output? You just have to multiply the heart rate with stroke volume. That is, if the heart rate is 72 and the stroke volume is 70, so nearby it will be 5040, I think 5040 ml blood. That will be the cardiac output. In lump sum we can say that is 5 liter. So that is the cardiac output, heart rate into stroke volume. That means heart rate is for 1 minute. So if for one stroke, one ventricle is pumping 70 ml of blood, so for 72 stroke, so within 60 seconds or 1 minute, within 1 minute of time, a single ventricle will pump near about 5 liter or 5000 ml of blood. So that is our cardiac output. So that is calculated for one ventricle, is not for both the ventricles. Right, now let's talk about ECG. Let's make it ECG. Electrocardiograph is the name of the machine. Electrocardiograph is the name of machine. And if you have visited a hospital or something like that, if a patient is going into the cardiac arrest, in the monitor, the patient is lying beside, and in front of that patient there will be a monitor which is measuring the electrical activity of his or her heart, and that is creating some lines that we are calling electrocardiograms, right? And once he or she is going to cardiac arrest, you may hear a sound like pip, 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 pip. Now the patient is dead, almost dead. So the graphical representation of electrical activities of our heart during the time of cardiac cycle, that is called electrocardiogram. So the graph which is produced during the time of cardiac cycle, that is the electrocardiogram, and the machine is called electrocardiograph. So the graph is gram and the machine is graph here, right? Now how this graph is produced? Okay, so you have to measure the electrical activity of our heart. We know that your heart has numerous nodal tissue. It has nodal tissue like SAN, AVN, atrioventricular node. It has bundle of E's or AV bundle, Purkinje fiber, right? So the heart has its own rhythmic components to produce, which can produce the depolarization or the action potential, a kind of electrochemical energy is produced inside our heart with the help of this SAN, AVN or bundle of E's or Purkinje fiber. So we can measure the activities of those electrochemical impulse when they are getting propagated or conducted with the help of some device that is called electrocardiograph. Okay, now how it works? See, some electrodes or leads are installed into our body. One lead that is negatively charged installed to our right arm, here the wrist of our right arm. Another lead which is of positive charge installed into our wrist of left arm, and another lead which is installed into the ankle of left leg, and so they are making a triangular structure, and we know this lead is also positively charged. Our heart is tilted towards left, right? We know very well. So that is making the apex a positive end and the base a negative end. So basically with the help of some device or some electrodes or leads, we will be measuring the potential difference while the electrochemical energies are passing through the nodal tissue inside our heart. And to measure the activities in a very close manner, near to the heart, 6 more leads are installed into this location, into our left chest region. We name them V1 to V6. They will be measuring the activity of heart in a very close manner. Okay, and after measuring, they will be giving us a graphical representation. When there is a straight line, then there is no change, no net change is taking place inside the electrochemical circuit inside our heart. Sometimes there will be a jump upwards, that is called positive deflection. Sometimes there is a dip downwards, that is called negative deflection, right? The straight line, we call it isoelectric line. When there is change of net charge or potential difference is 0. If the jumping sign is seen, that is called positive deflection. If the dipping sign is seen, that is called the negative deflection. And ultimately that will create a graph for a normal ECG, a normal person like this. Now let's elaborate this graph. So the graph starts with a wave called P wave. What is the importance of the P wave? When the electrochemical currents are flowing through the cardiac muscles of atrium. So that is defining P wave, that is defining atrial depolarization. Okay. Now isoelectric line, then there is a dip, that is called Q point, and there is a jump, and ultimately the top point it will form R point, and again coming down another dip, that is called S point, again a straight line. Okay, so this particular complex where you got a Q, R and S, we call it QRS complex, and that QRS complex defines ventricular depolarization. So depolarization means positive charges are flowing through ventricle. Okay, now after that, at the end of this straight line, there will be formation of another wave that we call T wave. Significance of T wave is ventricular repolarization. So basically, what do you mean by depolarization and repolarization? Depolarization means where there is formation of positivity, repolarization when there is formation of negativity. Okay. So P wave, that denotes atrial depolarization. QRS complex together they are denoting ventricular depolarization. T wave, that is denoting ventricular repolarization. Now you might be thinking that, sir, where is atrial repolarization? We have atrial depolarization, ventricular depolarization, ventricular repolarization, but where is atrial repolarization? It's so weak that somehow it's must, atrial repolarization is must within this QRS complex. Okay, remember, this graph is not showing us that how our heart is performing systole or diastole. It's showing us that what kind of electrical activities or charges, electrochemical signals are flowing inside through the nodal tissue of the heart. So sometime you say that depolarization means systole, repolarization means diastole. It's not like that. Depolarization is not equal to systole, repolarization is not equals to diastole. Remember this. Rather, we can say that systole is the consequence of depolarization, diastole is the consequence of repolarization. So first there will be depolarization, then there will be a time gap, and after that the heart will start performing systole. First there will be repolarization, there will be time gap, then your heart will start performing diastole. It's just like that. If you are running into a running competition, just at the starting line, you are waiting for the whistle, right? So first the whistleblower will blow the whistle, and there will be time gap of some seconds or milliseconds, then you will start running. So as soon as the time point, if you take the timeline, the whistle blow and you started running, that will not be in the same point, right? Similarly, first your heart will perform depolarization, then the message will reach to the cardiac muscle, then they will start systole. So somehow in the middle zone, middle point of this P wave, the atria will start its atrial systole. Somewhere the middle point of this QRS complex, your ventricle will start ventricular systole, and just this point somehow somewhere near to the ending point of the T, that is depicting or denoting the ventricular systole, and ventricular diastole will start after completion of the T wave. So T wave doesn't denotes, the starting of T wave will not be denoting the ventricular diastole. starting of T wave will be denoting end of ventricular, not end, most of the T wave is just ventricular systole. Again, I am repeating it. P wave means atrial repolarization, right? So just if you are thinking when the atrial repolarization will begin, the same time atria will start systoling, it's not possible. Okay, somewhere into the middle zone of P wave, the atria will start its contraction. If you think that just at the point of Q, the ventricle will start its contraction or ventricular systole, that's also not possible. Somewhere first this wave will start, the electrochemical signals are spreading throughout the ventricular musculature, and in the middle zone of QRS complex, somewhere the ventricle will start its contraction or ventricular systole will start here, and ventricular systole will end near to the end point of T wave. So most of the T wave, though T wave is for ventricular repolarization, so you may think that that is for ventricular diastole, but in NCERT is written that is for ventricular systole. Why it's so? Because first there will be completion of T wave, that is ventricular repolarization, then your ventricle will start for ventricular diastole. So this is how the graphs are formed. This is how a patient is observed by the cardiologist. So these are the leads. Right. Now I'll talk about double circulation. So double circulation means a four-chambered heart. If you observe a four-chambered heart, the oxygenated blood is received. Oxygenated blood is received and pumped by the left side of our heart, and the deoxygenated blood is received and pumped by the right side of our heart. The heart is successfully separating the oxygenated and deoxygenated blood. Okay, and for oxygenation, the heart is sending the deoxygenated blood to the lung. Lung is performing the oxygenation of the blood. The blood is converted into oxygenated from the deoxygenated blood into the lungs near to the alveoli, and that oxygenated blood will be circulated by pulmonary veins into the left atrium, right? So here this is one cycle from heart to lung and lung to heart. Now again, the left ventricle of your heart will pump the deoxygenated blood through the systemic artery that will bring the oxygenated blood to the tissues, donating the oxygen and gaining the carbon dioxide. The blood will get converted from oxygenated to deoxygenated into the systems or the organs of your body, and your veins or vena cavas will be collecting the deoxygenated blood from the organs and bringing back to your right atrium of your heart, right? So heart is connected in one way, your heart is connected to the system. In another way, the heart is connected to the lungs. So the connection to the lungs, that is called pulmonary circuit, pulmonary circulation, and the connection of heart to the organs or system, this is called systemic circulation, right? Pulmonary circulation start from right atrium. Pulmonary circulation start at pulmonary circulation start from left ventricle through the pulmonary artery is delivering the deoxygenated blood to the lungs, and lungs are pumping back the oxygenated blood through pulmonary veins and the pulmonary veins are connected to the left atrium. So this is your pulmonary circuit. Now if we talk about the systemic circuit, it will be starting with left ventricle. From left ventricle to systemic artery is connected to the organs or the systems of our body. From organs, the deoxygenated blood will be collected by systemic veins or vena cavas, and the vena cavas will open into right atrium. So from left ventricle to right atrium, this is the systemic circuit, and from right ventricle to left atrium, this is the pulmonary circuit. From right ventricle to left atrium, pulmonary circuit. From left ventricle to right atrium, this is systemic circuit. Okay, so this was about double circulation, and also here we need to talk about the blood vessels of different diameters, that is arteries, veins and capillaries. Right. So for arteries, what you can observe, see, for arteries and veins, they are made up of 3 layers. And capillaries, they are very smaller. They are ultra thin in nature. Okay. The middle layer of arteries is made up of smooth muscle and fibrous tissue. Similarly, in veins, so arteries and veins, they have 3 layers. Arteries and veins, they have 3 layers. Okay. So what are the name of those 3 layers? The outermost layer, that is tunica externa, middle layer tunica media, inner layer tunica intima. Similarly with veins, the outer layer tunica externa, middle layer tunica media, inner layer tunica interna. Now you please observe here the thickness of tunica media in between artery and veins. The tunica media is thicker in case of arteries, and that's why the lumen of artery is narrow and lumen of veins, that is broad. And when you talk about capillaries, they're just made up of one single layer of squamous epithelial cells. So they are ultra thin. Okay. Now what is the function of arteries? They will be carrying blood from heart to organs. That is true for all the arteries. For all the veins, it's true for they will be carrying blood from organs to heart. Exception pulmonary veins, they are carrying oxygenated blood. For arteries, exception is pulmonary artery, they are carrying deoxygenated blood. And for capillaries, basically they are present inside organs. They connects or they links arteries to veins. Right. The maximum surface area of blood vessel is contributed by capillaries. The maximum surface area is contributed by capillaries. The blood is flowing at a high speed, high velocity through the arteries. So the velocity of blood is high in arteries. The velocity of blood is low in compared to arteries into veins. Okay. So as because the blood, whatever flowing through the arteries at a high velocity, there is no chance of the backflow. But in case of veins, as because the blood is flowing into very low relative velocity, so to prevent the backflow, the inner wall of veins are provided with valves. So the inner wall of veins, you will be finding valves. So valves are present inside throughout the walls, inner wall of veins, but inside the arterial wall, valves are absent. Right? Now let's move to the next topic. We'll be talking about hepatic portal system. So what is hepatic portal system? See, so in normal cases, for normal cases, like for other systems except the hepatic portal system, the deoxygenated blood is collected by the veins, and the veins will open to the highway of veins, that is systemic vein or into vena cava, right? So for normal organs, the deoxygenated blood is collected by the veins, and veins will be opened into the or released into the vena cava, that is the highway or the main vein of our body. One more thing I am telling, suppose there are two houses, house A and house B, and in front of those two houses there is a highway. Now the person who is living into house A directly getting a road which is leading him towards the highway, but the person who is residing in house B don't have the direct road towards the highway. So basically what is there, he has constructed a roadway which connects him to the roads in front of the house A. So the person of house A can directly reach to the highway, but the person which is residing in house B, he or she has to first come into the road of house A, and then this road will lead him or her towards the highway. So here basically the road of the person A is providing a gateway or port for the person B. So this system we can call as a portal system. Okay. Similarly, into our body, the veins of our gastrointestinal tract or digestive system, like pancreatic veins, the veins from stomach, the veins of small intestine, they are unable to join directly with the vena cava. So the deoxygenated blood through the veins of these particular organs of our digestive system will be thrown first inside the veins related to the liver. So the hepatic veins are acting as the port for the veins of these organs related to our digestive system, and ultimately the hepatic veins, through the hepatic veins, the deoxygenated blood of those organs are coming into the inferior vena cava or the prime or the principal vein of our body, that is systemic vein. So that's why we are telling that here the veins of liver, that is hepatic veins, are acting as portal. So this system is called hepatic portal system. So I am summing up the things. The veins form digestive system, they're unable to throw their deoxygenated blood directly into the inferior vena cava, so they have to use the way of hepatic vein. They are getting merged with hepatic vein after some time, and the hepatic vein just helping the veins of these organs to deliver the blood into inferior vena cava. So this is called hepatic portal system, when the hepatic veins are giving port to the deoxygenated blood or veins which are coming from the organs of digestive system, and via liver they are getting opened, or the blood is getting released into the prime vein. Okay. Now we'll be talking about the regulation of cardiac activity. The regulation of cardiac activity. See, we know that heart is auto-regulated organ, or heart is myogenic. The heartbeats are produced by sinoatrial node or SAN. Okay, but modulation or moderation of heartbeat can be done with the help of some nerves. It's called neural moderation, some part of our nervous system, and obviously with the help of some hormones. So let's talk about neural moderation of our heartbeat. So we know that we have autonomic nervous system, ANS. This autonomic nervous system has two subtype, sympathetic and parasympathetic. Remember, this sympathetic nervous system always increases heartbeat and cardiac output. Parasympathetic nervous system always decreases heartbeat and cardiac output. Okay. There is another neural control over our heart, that is the vagus nerve. So this vagus nerve that also can slow down the cardiac activity by giving negative feedback. So vagus nerve that also decrease cardiac activities. Now let's talk about the hormonal control or hormonal moderation. Okay. The hormones of our adrenal medulla, that is adrenaline and noradrenaline, remember, they will always be increasing the cardiac activity, as well as the thyroid hormones. They also help us to increase the cardiac activity. So the cardiac activities are boosted by sympathetic nerves, by adrenaline and noradrenaline hormones, by thyroid hormones, right? And also, whenever we'll be studying the chapter excretory product and elimination, there we're gonna get to know about a cycle or a mechanism of controlling our blood pressure into our body and the kidney filtration, that is called RAS system, renin, angiotensin aldosterone system. So that RAS mechanism also helps us to increase our cardiac output and blood pressure, and the opposing one, that is atrial natriuretic factor, that helps us to decrease the cardiac output and blood pressure. So for this chapter, this information is enough. Heart is showing autoregulation by SAN and the nodal tissue. When you talk about neural moderation, autonomic nervous system can moderate the heartbeat, either they can increase or decrease. When autonomic nervous system can increase the heartbeat, when the heart is under control of sympathetic nerve. When the heart is under control of parasympathetic nerve, the heart rate and the cardiac output and heartbeat will be decreased. Even the stimulation of vagus nerve, that can decrease your heartbeat or cardiac activity. Now the hormones we have talked about, adrenaline or epinephrine, noradrenaline or norepinephrine, as well as thyroid hormones, they have positive effect on cardiac activity. They can increase cardiac activities. Now let's talk about some disorders. So first we'll talk about the hypertension. Hypertension means high blood pressure, high BP. So what is normal blood pressure? 120/80. Systolic pressure, the upper one 120 mm mercury pressure, diastolic pressure into the arteries, that is the 80 mm mercury pressure. That is the normal blood pressure. Now, if repeated check of blood pressure of an individual is 140 or 90 or even higher, then that condition can be concluded by saying that person is suffering from hypertension or high blood pressure. And if you are suffering from this chronic high blood pressure, that gonna affect your kidney and organs like brain. See, generally, if there is continuous high blood pressure for a long time, the doctor will be suggesting you for a kidney checkup, because as I said, there are certain factors or hormones produced by our kidneys. They are really responsible to maintain our cardiac activities. Okay, now we'll talk about coronary artery disease, another disorder that is called CAD, coronary artery disease. Another name of this disease is atherosclerosis. So what happens during the time of coronary artery? Coronary artery, so heart, the left ventricle of heart or the aorta supplies blood to all the organs, but it's the coronary artery, some certain branches originating from the aorta, right? There are few branches which are originating from aorta, the base of the aorta. They will be supplying oxygen to the heart's own muscles, so they are called coronary arteries. So coronary arteries, they are responsible for supplying oxygenated blood to cardiac muscles. Now, if there is deposition of calcium, fat, cholesterol and fibrous tissue inside, then they will form a plaque. They will form a lump that we are calling fatty plaque inside the lumen of coronary artery, and ultimately that will be blocking the supply of blood towards the cardiac muscle, and that can be cause of many other diseases. So this condition we call it coronary artery disease or atherosclerosis. There is another disease synonymous to this atherosclerosis, that is arteriosclerosis. When small arteries, the wall of small arteries are hardened by deposition of fibrous tissue and calcium, that is called arteriosclerosis. So this is atherosclerosis. The main villain is here calcium, fat, cholesterol and fibrous tissue. Okay, that's making a blockage towards the blood flow. And for arteriosclerosis, that is the hardening of the arterial wall mainly. Okay, now another condition, angina pectoris or angina, basically that is a form of chest pain, acute chest pain, sudden chest pain. Why? If there is no enough oxygen supply to your cardiac muscle. It may be due to the consequence of the atherosclerosis, due to the consequence of CAD, right? So this is more common in middle-aged and elderly human, is because with age the plaques will be developing into our coronary arteries or the important arteries, that can cause this angina. Though it can be observed like in human of any age, but more common in middle age and old women, right? So how you can define it? Angina, the acute means sudden chest pain. Why it's happening? Because your cardiac muscles are not getting enough supply of oxygen. Okay, now this angina can lead to heart attack or myocardial infarction. Okay, there is another disorder, heart failure. So what is the difference between heart failure and heart attack? First, we will be talking about heart failure. When heart is not pumping blood efficiently, we know that ventricles are pumping 70 ml of blood in one stroke. So if there is ventricular fibrillation or ventricular dilation, in that case heart chambers won't be pumping the blood in a proper manner. Heart is failing to supply optimum amount of blood towards our body tissue. That's called heart failure, and sometimes that can create congestion. Into our lungs. So if there is congestion into our lungs due to the heart failure, that is called congestive heart failure. Now why it happens? See, when the ventricles are failing to pump enough blood, then obviously from the previous cardiac cycle, already blood is stored inside the ventricle. That will create a backflow pressure. That will create a backflow pressure towards the lung. That will create a backflow pressure towards the lung, because it's the lung which is sending oxygenated blood with the help of pulmonary vein to the left atrium, and ultimately the left atrium is pumping blood to the left ventricle, and left ventricle is pumping blood throughout the body organs. And if the left ventricle is not working properly, from the source, that is from the lung, things will be disturbed, and lung will, you can observe accumulation of fluid, the excess blood which is getting stored inside the lungs. Lungs are unable to pump or unable to send them through the pulmonary veins towards your heart. They will create problem, they will create congestion into your lungs, and that we call congestive heart failure. Now, what is heart attack? Heart attack or myocardial infarction or myocardial ischemia. The three terminologies can be asked, but the meaning is same: heart attack. Now, what happens here? These are not death of your heart, but damage of your cardiac muscle. Why cardiac muscles are getting damaged? Because there is inadequate blood supply. Maybe the patient was suffering from high blood cholesterol, that CAD, coronary artery disease, and that may lead to damage of few cardiac muscles, and that we call heart attack. But yes, heart attack generally don't kill a patient, but if there is massive heart attack, that can kill, and heart attack can also give rise heart failure, right? So, precise difference between heart failure and heart attack. In heart failure, ventricles are not working properly. In heart attack or myocardial infarction means kind of damage, right? So, when few cardiac muscles are dead or damaged due to inadequate blood supply, because the cardiac muscles are not getting enough amount of oxygen and blood, so they will start dying. That is called heart attack. And the third disorder, that is cardiac arrest. Now, cardiac arrest means there is a full stop of heartbeat. Heart completely stops beating. Now, maybe the patient will die. Okay. Now, the ECG graph will not show any deflection. It will just show the straight line, the isoelectric line. So, that denotes cardiac arrest when heart completely stops beating. So, that was all about the cardiac disorders. Now, let's solve this question. One statement is saying that heart, the endodermally derived organ, is situated into the thoracic cavity. So, it's never endodermally derived, heart is mesodermally derived. So, the statement 1 is false. Statement 2 is saying the walls of ventricles are much thicker than the walls of atria. Yes, obviously, ventricles are blood pumping chambers. With great power comes great responsibility. Why they are stronger? Because they are more responsible. In the light of the above statements, choose the most appropriate answer. So, statement 1 is incorrect and statement 2 is correct. Now, let's solve this question. Which of the following will never carry deoxygenated blood? Okay, few blood vessels examples are given here. Okay, a renal vein, will they carry deoxygenated blood? Yes, they will be carrying deoxygenated blood. Hepatic veins, yes, they are carrying deoxygenated blood. Pulmonary artery, will they carry deoxygenated blood? They are exceptional arteries. They are only a artery, pulmonary artery, only artery to carry deoxygenated blood. They are carrying blood, deoxygenated blood from the right ventricle to the lungs. Now, if you observe this name, pulmonary veins, yes, they are the arteries responsible to bring oxygenated blood from lungs to left atrium. So, always they will be carrying oxygenated blood. So, this is our answer. They will never carry deoxygenated blood. Okay. Identify the type of circulation given in the diagram and choose the correct option. So, the diagram is showing heart pumping deoxygenated blood to gills, and gills pumping oxygenated blood to rest of the body, rest of the body is pumping deoxygenated blood to heart again. So, this is a circulatory pathway. There is single circulation observed in fish. So, this is single circulation. So, in fish what happens? Body organs, they will send deoxygenated blood to heart, heart will receive deoxygenated blood, heart will pump deoxygenated blood. That is why single circulation. And the deoxygenated blood will be sent to gills, and in gills there will be exchange of carbon dioxide and oxygen, and in gill there will be formation of oxygenated blood that will be sent to rest of the body. Perfectly, this diagram is denoting towards the circulatory system or circulatory pathway of fish, that is single circulation. Okay. Next question, doctors use stethoscope to hear the sounds produced during each cardiac cycle. Yes, the second sound is heard. What is this? What is second sound? That is dub, right? When semilunar valves close down after the blood flows into the vessels from ventricles, or when AV valves, okay, every time when valves will be closed, sound will be produced. So the second option, when the valves are opened up, there will be no sound. Ventricular wall vibrates due to the gasping of flow of blood from atria. This can't be the reason. Ventricular wall cannot produce sound. AV node receives signal from SA node. When signal is getting transferred, that cannot produce sound. So the answer is closure of semilunar valve. Remember, in the beginning of this lecture I said, when you see the shining moon, that is Luna, you will listen dub. So closure of semilunar valves will be producing dub sound. And why semilunar valves are closed? Just to prevent the backflow of blood from the arteries into the ventricles. So this was the end of this particular lecture. Hope you have enjoyed, and I have tried to elaborate the topics in very detailed manner. So we are also coming with, in coming days, with individual lectures, like our chapters will be covered in detailed manner with proper PYQs and question practice. And for the timing, please stay tuned. Again, please like, subscribe and share our channel. There is PW NEET English, and for the timing, I am saying Jai Hind, bye bye.